It wasn't until my fourth year when I was taking a course taught by Dr. Jane Yak. And that course was sort of like an introduction to the diversity of insects. We had to go outside in the city and collect 14 different orders of insects and sort of like pin them in a box and then hand that in. I don't know why I enjoyed it, but I kind of liked going out there and seeing like where these insects live and exactly like how to preserve them, whether to like pin them or, some insect, for example, you can't pin them, you have to put them in a bottle of alcohol.
What really got you interested in working with fruit flies?
Fruit fly anatomy is very simple relative to humans, I guess, because, they do have a brain like us, and they do have a tracheal system, kind of unlike us in the sense that we have a windpipe, we have lungs, right? But their tracheal system kind of extends towards the organs directly. They don't have blood vessels like we do. Their circulatory system is open in the sense that all the organs are sort of bathed in this fluid called hemolymph. Their fat body is kind of like the analog to our liver, you can think of it. All these organs are bathed in this hemolymph at all times. And what we know about damage that happens in fruit flies has been looked at in the gut, and we know that the gut becomes damaged as a result of cold stress.
You're trying to figure out how tolerant they are to cold temperatures and understanding how their immune system is correlating with that cold tolerance. Why can't they, or how do they tolerate cold? What's the mechanism they use to do that? Mahmoud, how's it going?
Going well. How are you, George?
Good, thank you. Thank you for coming on the podcast.
Yeah, thank you for
I
having me.
appreciate it. We had a chat, a couple of weeks ago?
Yeah, we
It
did.
was, yeah, over some coffee.
Oh, yeah.
We always run into each other, and we
Oh,
say
yeah.
hi, but I don't think we were introduced formally to each other. Yeah, you work in Dr. Heath MacMillian's lab,
Yeah.
and I want to know a bit about your background, your story. How did you get into research, and what really got you interested in working with fruit flies?
Ooh.
So I'll let you take it from here. Give us a little bit of background about yourself.
All right. So I first started my undergraduate degree here at Carleton in 2013.
Mm-hmm.
And at that point, I didn't have a solid idea of like what I wanted to do at university. I was kind of like going with the flow. I had big sort of like visions of me being in medicine to some degree. I don't know if it was like nursing or like some sort of surgery, but I wanted to go something medical focused, right? And it wasn't until my fourth year when I was taking a course taught by Dr. Jane Yak. And that course was sort of like an introduction to the diversity of insects. It was a biology course, and I needed some upper year biology courses to like fulfill the requirements of my degree at that point. But I never studied insects generally before. And I thought, hey, this course seems cool, why not? And something that we had to do in that course was go out. This course happened from September until December, so weather was still warm, and we had to go outside in the city and collect 14 different orders of insects and sort of like pin them in a box and then hand that in. And that was sort of our final assignment
Mm-hmm.
for the course, which was worth about, I think, like 25% of our final mark.
Okay.
And it was really neat. And I don't know why I enjoyed it, but I kind of liked going out there and seeing like where these insects live and exactly like how to preserve them, whether to like pin them or, some insect, for example, you can't pin them, you have to put them in a bottle of alcohol.
Can you explain the difference between pinning them and putting them in a bottle of alcohol for those that don't know,
Yeah,
including
so
myself?
yeah, so some insects are relatively large. I say relatively is because, if you look at something like a fruit fly that's like 2 to 3 millimeters, right? But then you have some ladybugs, for instance, that are close to a centimeter big, right? Those insects are relatively larger. And you can take a ladybug or any beetle and sort of like pin them in such a way that it penetrates their thorax,
Mm-hmm.
and you can pin them to the bottom of a collection box. So you have this sort of insects like pinned halfway between like the needle and the bottom of the box, and you can also attach a label that has the order of the insect, date it was collected, location it was collected, right? And this is how taxonomists or anyone curating insects, this is how they typically preserve them.
Okay.
Some other insects are very, very small in the sense that you can't really pin them quite well, because of their size. You try pinning them and you just end up destroying the whole thing.
Like what? Like ticks, for example?
Yeah, ticks or other orders such as Archaeanatha, for instance. They're like these really small insects that have these hunched backs, but their exoskeleton isn't quite tough enough to be pinned.
Okay.
So they're very like small, they're very delicate. In that
Yeah.
case, you have to take a bottle of alcohol,
Mm-hmm.
and you have to sort of like dump the insect in there. and attach a label to the outside of the container with the same details as well.
Can you use formaldehyde? Because I know that some labs use formaldehyde. What's like the difference between that versus alcohol?
I'm not 100% sure on the difference between the two. Typically, ethanol is used as a preservative for curating insects.
Okay.
For the purposes of our course, I'm guessing we weren't able to use formaldehyde because of the hazard associated with the chemical.
I see. Yeah.
And ethanol is good enough. All you need is like 70% ethanol, right? And just like any sort of like medicine holders, the one they give you at pharmacies, the
Yeah,
little
like the
orange
little
containers. Yeah.
the orange containers.
Yeah.
The cylinder one, like the
Yes,
yeah,
exactly.
cylindrical
So
ones, yeah.
I didn't need like special containers. I just had one of
Right,
those
okay.
sitting at home. I just like, emptied
Yeah,
it out,
yeah.
dumped the insects in there, and just called it a day.
Okay. And whatever alcohol that's left, well, hey, that's a free drink right there.
Exactly.
Yeah.
Cheers after insect collection. Well done, I guess.
Yeah. Okay, so then the two different methods, like you said, are pinning,
Mm-hmm.
primarily two different methods, pinning and using a like alcohol, for example,
Yeah.
as a preservative.
Right.
So when you're pinning the insects,
Mhm.
how do you do it for one? And like, do you have to do through the wings, through the legs? What
Mhm.
parts of the insects are you selecting to pin for one? And two, the box that you're putting it in, is it like sealed? Is it vacuum? Is it vacuum sealed so that you don't have any air inside?
Yeah, so the boxes are typically sealed for
Mhm.
one. So insect collection boxes typically have a snap lid,
Mhm.
where it's just a simple lid that sort of like has a hinge and it closes in on itself with a little lock at the front of it. And no air gets inside the box. So you have all your insects in the box, and they're typically like away from the natural elements. The pinning is typically done through the wings. So typically, insects have two pairs of wings, right? If you're looking at something like beetles, for instance, where one pair of the wing is very hardened and they're very tough, right? If you take the insect pin and go directly in the middle, you kind of like miss both wings, and it's hard for the insect to stay affixed to the needle.
Right. Okay.
So it tends to like slide down.
Mhm.
So the trick is to actually go a little bit to the right or the left and go through one of the wings in the middle of it. So sort of go like laterally in a
Okay,
sense.
okay.
So typically when you see insects being pinned, they're typically like aligned to the left or aligned to the right for
Mhm.
that reason.
I see. Interesting. Okay. So that's the first course where you had a love for insects, or you found a dedication to curating insects.
I think that was sort of like my gateway
Mhm.
into the insect world. Yeah.
Okay. And then what made you decide that once I'm done with my undergrad degree, I do not want to pursue medicine. I want to actually go into graduate research and work with insects.
So my first step into the research world happened during that course, actually.
Okay.
It was kind of a weird turn of events. So we were learning about insects, right? And we were getting used to identifying insects based on distinguishing characteristics, like how do you distinguish a ladybug from a mosquito, for instance, or how do you distinguish a fly from like a termite, right? And at some point, Jane sent out an email to the class saying that there was a master's student, Lauren Moretto, who was in one of the landscape ecology labs upstairs here on campus in the biology building. And she went to Toronto. She was doing bat distribution surveys, looking at the abundance of bats. And she collected a bunch of insect samples with her back. She sort of like went out into the brush, collected a bunch of insects, brought them back, but she was looking for people to identify them.
Okay.
And I think the idea at that time was to relate the abundance of bats in Toronto, in the urban areas, to the biodiversity of insects in those areas. So let's say you find like these species of insects here. Does that affect the distribution of bats in that area? Like, can we correlate bat activity to whether we find these insects here or not, right? And identifying the insect was step one. But, Lauren was very proficient at the landscape ecology stuff, but us being an insect course, she was looking to recruit. So she sent out an email. And since I was already like in that zone, I guess, halfway through the semester looking at insects, I thought, hey, why not? Might as well, right? So I replied to her back saying I'd like to help. And that was my first experience in a research lab outside of my undergrad courses. Because up till that point, everything that I did in my undergrad courses was just limited to going into the lab like 3 weeks at a time.
Okay.
Right?
Yeah.
Where we go into the lab, we have some sort of objective that we need to do, right? And then once we do that, we don't go back into the lab until the next week, right? And we're being graded on it, and we're writing reports. But this is sort of like a bit more open-ended because, I wasn't limited to 3 weeks at a time.
Right.
I
Right.
had a lot of time in terms of like volunteering for Lauren and stuff.
Mhm.
And I was doing that for close to a year.
Wow. Nice.
Yeah.
Okay. After that point, when did you decide that you wanted to get into graduate research?
So when I was at the year mark of volunteering with Lauren, I heard news that we were getting a new faculty member, Heath MacMillian. At that
Mhm.
point, he was finishing up his postdoctoral work at York,
Okay.
and he just got the position of an assistant professor here at Carleton. So I went on to the university website, right? I saw his profile, and I saw, oh, I work with low temperatures and insects. I was like, oh, well, that's fantastic. I'm already working with insects right now, right? With Lauren. And I thought, well, I want to get more exposure to working with insects. So I shot him an email, and being a new prof, I asked him like if he was looking for volunteers to like, help set up the lab and everything like that. And he was more than happy to take me on. Me being new to relatively new, I guess, to the research world, and him being new to campus, I think it was sort of like a coincidental meeting between the two of us. It was
Mhm.
very good timing where, I was looking to get more involved, and he was also looking for people to like help him around. And
That perfect place at the perfect time kind of deal.
exactly,
Yeah.
yeah. It was
Yeah.
kind of like serendipitous in a sense,
Yeah. Yeah.
And, it was just me volunteering, helping him out with a lot of like tasks in the lab, like, unpacking boxes, taking inventory.
Mhm.
Any of the other honor students or his master student that needed help, I was, helping him out with like taking care of the flies and things like that.
Okay.
And partway through, after a few months, he asked me if I had an honors thesis supervisor, which I didn't at that time because I was like in my last year. And he said, well, if you're looking to do an honors with me or like a directed studies, like an independent project, you just let me know and we can work something out. And
Sweet.
I didn't have to think too hard,
Yeah.
honestly.
Yeah.
And I talked to him the next week. I took him up on it. And yeah, I started my first independent project in the winter of 2018.
And now, as they say, the rest is history. You're in your PhD right
That's
now,
right.
right?
Yeah,
And you had your comp exam
I
last week, you
did.
said,
Yeah, I did.
on Friday. How was it?
It was it was challenging. It was challenging in a way that it didn't quite occur to me how much I still don't quite know in my field, if that
Mhm.
makes sense.
So, can you just explain to the listeners what
Mhm.
a comp exam is and why PhD students have to do it?
Yeah, so the comp exam is as the name states, it's a comprehensive exam that a lot of universities have for its doctoral students. So typically when somebody starts their PhD, right, they get into the program, and within a time frame that kind of differs by the institution. Here at Carleton, it's about 18 months. So within 18 months of first starting your program, you're expected to do the comprehensive exam. And the point of the comprehensive exam is to determine whether the student has a solid understanding of their own project and whether their thesis, looking at the chapters as a whole, sort of has a logical flow to it and
Mhm.
would be um, whether it would make sense to sort of like do that project in the first place.
Okay.
Looking for any not sort of like flaws, but any like logical holes, for instance,
Mhm.
right? And whether the candidate knows how to approach research in the sense of setting up experiments and understanding what they're working with. If they're working with animals, do they understand the animal that they work with?
Yeah.
um Analyzing data and just having an overall good understanding of their field. And I had mine this past Friday.
Okay. How'd it go?
I think it went pretty well. And the reason I say I kind of like found it challenging at first is because the comprehensive exam committee doesn't always involve professors that are familiar with their work.
Mhm.
So, I work with low temperatures and insects, for instance, right? But I had a couple of professors that don't work with insects at all. They work with like proteins or cell culture. So, when they ask me a question like, oh, you work with flies at low temperatures. Do flies even feel low temperatures? Like, how do they do that? Yes, questions like that doesn't really like come naturally.
Yeah. Yeah.
If you're
It's
like so zoned into
yeah.
your own work, right? Trying to explain that to somebody else who's coming from the outside, I guess it's part of like science communication, but it still has its own like little charm to it.
Yeah, sometimes it's the simple questions that aren't
Mhm.
very complex
Yeah.
in your field
Mhm.
or in the way the professor asks it. It
Yeah.
really makes you think like, yeah, why is that? So actually, what did you
Yeah.
answer that
Yeah.
for that question?
So Well, one of the questions I got was, well, my work is about insect immune systems, for
Mm-hmm.
instance, right? And one of the questions I got was, well, my this is coming from the professors. He was saying, oh, like, I don't work with insects. And when I was reading your proposal and just reading that Drosophila melanogaster fruit flies, just reading that Drosophila have an immune system kind of surprised me because I thought as humans, we are the only ones that have that, right? Mice are mammals that they have an immune system as well, but insects, they're this small, right? 2 to 3 millimeters. How do they have an immune system? What are they fighting off, right? And then he asked me like the difference between an insect immune system and animal and a human immune system. Like how are they similar? How are they different? Right?
Those are some good questions, actually. I'd
Oh
ask
yeah.
you the same thing, to be honest.
It's a really good question. As you said, like the simplest questions that you don't really like expect, right?
Yeah.
But they're usually the hardest hitting ones because at its
Interesting. Yeah.
foundation, right? When
Yeah.
you're looking at some sort of system that you're studying,
Mm-hmm. Mm-hmm.
how do you relate the system to something else that's already well known? A lot of the human immune system is relatively more well understood than something
Right.
like insects because we're still looking at that.
Yeah. Yeah.
And that's only been described only in the past 2 decades.
That's interesting.
Hmm.
Okay, so I want to dial it back a little bit and talk about your work. Give me a comprehensive overview of what you do in your PhD currently. What's your hypothesis about?
So my current project right now is looking at insect immune systems at low temps.
Mm-hmm.
And the gist of it is that insects don't tend to do well when temperatures go very, very low, right? They're ectotherms. They can't regulate their body temperature. So if it gets too cold outside, their body temperature also goes down and they can suffer injury to their body, right?
Mm-hmm.
And what's sort of like been described over the past decade or so, it's relatively new, is that their immune system also activates at low temperatures as well. Even in the absence of any bacteria, any fungi, or anything like that, certain immune genes in their body are overexpressed following that low temperature stress,
Okay.
and we don't quite know why that happens.
Mm-hmm.
So my research right now is trying to establish those mechanistic links between cold exposures and immune activation in insects, and I'm using the fruit fly Drosophila melanogaster as my model organism.
Okay, fruit flies. We're all too familiar with that. Leave a banana out for a little too long or something.
yeah.
Actually, a question about that because I'm curious. So sometimes I will leave a banana out, forget about it, and then fruit flies will start to populate. Where are they coming from originally? So because I
Yeah.
don't find them around. Like they're not just chilling around my house and they
Yeah.
start to lay eggs in the banana. Are
Yeah.
they coming out of it, out of the banana?
There's always a possibility that some eggs might find their way into the fruit itself, but fruit flies are very small as well, right?
Yeah.
They're like 2 to 3 millimeters large.
Mm-hmm.
And if there's any opening, whether it's in the kitchen or anything in the house that has an
Okay.
opening that big, right? They can just squeeze their way in and
Okay.
find their way.
Okay.
And they can sense the presence of any fruit, like fruits or anything sweet, for instance, sugar. They're able to pick that up and they're able to find that location. So
How do they sense that there's a fruit nearby?
So the literature right now is a bit like we're still trying to figure out what exactly they pick up in terms of compounds, but it's usually volatiles. So any volatile compound that emanates from that food source and finds its way into the air, they can usually pick up on those and they can direct their way to that food source.
Interesting. Okay. All right. So then you mentioned two things there. You're trying to figure out how tolerant they are to cold temperatures
Mhm.
and understanding how their immune system is correlating with that cold tolerance, right?
Yeah.
So let's kind of break down the first half. For cold tolerance, how tolerant are they? Because I don't find flies during the winter most of the time. They're mostly in temperate, warm climates, right? It's like in the summer or spring. So why can't they, or how do they tolerate cold if they can? And what's the mechanism they use to do that?
So talking about fruit flies specifically, they're not what we call chill tolerant in the sense that even when temperatures go down to at a point where it's still quite above the freezing point of their body, they still can suffer injury to their tissue. And the term that we use to describe insects like these are chill susceptible, where they're susceptible to the effects of chilling, but they can't tolerate it very well.
Okay.
So, Drosophila, the origins of Drosophila originate from Africa, and they're considered a tropical species. But, wherever they are on the continent, wherever they are around the world, when temperatures start to go down, they tend to migrate towards places where it's a bit warmer in order to try to survive that cold spell, right? But in a lab setting, where we have flies, being reared in a very controlled incubator, always set at 25 degrees, their optimal temperature, for instance, and we just put them at a temperature like 0 degrees, for instance, right? Because that temperature is still too cold for them, not quite freezing, but still cold enough, they can enter a state called chill coma, where they become comatose. So if you cool down an insect like Drosophila for long enough at a sufficiently low temperature, they lose the ability to move. They just kind of like slow down and they just plop on their sides and they become comatose.
And so, can they recover from that comatose state?
They can recover if they don't die from the cold exposure. So let's say, for example, staying at 0 degrees for a day is too stressful for the fly and it would die, right? It can't recover from chill coma. But if it stays at 0 degrees for something like 10 hours, for instance, where it's not quite stressful enough, it may suffer enough damage to its body or a bit of damage to the tissue, but it won't die outright. And you pull it out and plop it on a bench at a warm temperature, it'll regain the ability to move little by little, and it can stand up again, and it can actually recover from that chill coma state.
Is there damages happening when it's in that state, like to its body?
The damage that happens to the insect usually happens during the cold exposure, not following the recovery period afterwards.
Oh, okay.
So as long as it's suspended in the cold, it's going to accumulate damage to its muscle tissue as long as it remains in the cold environment. But once you pull it out and it doesn't die outright, then it will start recovering. It will be injured to some degree, and you can observe these injuries based on how well the insect is moving. So a healthy insect is able to like fly, it's able to like climb the walls of the container, it's able to jump, but a fly that's injured may not be able to fly, for instance. It may not be able to climb, and you can see its movement is very uncoordinated in a sense.
Can they repair any damage that happened during the point when they were in that coma state?
So what we know about the damage that happens is that whatever damage sets in during the cold exposure, the severity of that doesn't significantly increase or decrease during that rewarming period. So whatever tissue damage does happen in the insect's body, it tends to stay at that extent, essentially. So there's no repair process is quite happening, because whatever cells die, for example, in the muscle tissue, whatever cells die, those cells are going to remain dead. And the insect does not repair those tissues to a significant degree.
Okay. But I imagine they just replenish them, or do they not replenish them at all?
So one thing that we're trying to look at is how the tissue damage that sets in during the cold, how that translates to the whole organismal level in terms of their survival, right? Because there's already tissue damage setting in, right?
Yeah.
But, you look at the 24-hour period following that cold stress, where the insect does recover from chill coma, but there's a bit of degree of injury setting in, but the insect is still not fully at 100%, right? Does the insect die eventually? Does it remain at that level of injury for its whole life?
Okay.
That's sort of like the gray area that we have right now, right in our field, right? Trying to relate the extent of muscle tissue damage at a physiological level and going back to the organismal level and correlating those two things.
What's the comparison? I mean, I know they're completely different organisms, but what's the comparison between the types of muscles that insects have compared to what humans have, for example, or mammals have?
So insect muscles are largely involved in a lot of their locomotion, like wing movements and leg movements and things like that. Depending on what insects you look at, whether it's like fruit flies or locusts, there's going to be some comparative differences in terms of their anatomy, right? But, we don't have wings, for instance.
Right, yeah.
But insects do have wings as well. And there are muscles involved in the movements of their wings for flight, for instance. And whatever happens during the cold in terms of like what muscles are injured, typically studies that look to cold stress insects and look at their cold tolerance, they typically administer it to the whole organism. So they put the whole insect in that low temperature environment. Where that muscle injury is happening in flies, we know that it happens in their gut.
Interesting.
So the gut of fruit flies becomes injured at low temperatures. If you look at locusts, for instance, locusts are also chill susceptible, just like flies, in the sense that it doesn't have to become near freezing for locusts to become injured to low temps. And they also enter into a comatose state, chill coma, and they also recover from chill coma as well. And what we know from locusts is that the damage also happens to their hindgut, so the posterior region of their gut towards the rectum, but also from another muscle near their dorsal side. It's called the posterior pterygocoxal muscle.
Okay.
And that's typically the muscle that is easy to look at because it's relatively large. You dissect the locust from the ventral side, you can take that muscle out and you can stain it to look at live or dead cells. So that's how we know that muscle tissue is being damaged in locusts from all these studies using locusts as that model organism that made it easy to see.
All right. So then the next point I wanted to move to is how do you actually subject them to cold temperatures? What do you do? How does the process work? And how do you tally the results?
So I will mention upfront that there are different methods of looking at cold tolerance in insects. So what I mentioned about survival, right? Survival is usually the most straightforward metric. So you take some insect of your interest, let's say whether it's a fruit fly or a locust, right? You put it at a low temperature that's set, and then you look at whether that insect survives after 24 hours. We can call this a mortality assay. So you look at the ratio of live insects versus dead insects after that cold stress. And the way cold stresses are done, in our lab at least, they involve these big circulating cooling baths. So we have these cooling baths that we fill up with ethylene glycol or windshield wiper fluid.
Yeah.
And
just from the local grocery store?
Yeah, exactly. You just pick up a gallon or you're good to go. And you fill up the cooling baths with the fluid. And you can program the cooling baths to go down to whatever temperature you want. So let's say 0 degrees or -2 degrees.
That's the range that you normally treat them in?
So
what's the lowest you would go for a treatment? Or just a range is fine.
Yeah, so range-wise, it kind of depends on your question, but typically what we do with fruit flies, we've used anywhere from 4 degrees all the way down to -2. So that's a range of like 6 degrees Celsius. And fruit flies typically freeze, at least the population that we have, they freeze at around like -6, right? So once a fly freezes at -6, then it's pretty much guaranteed to die, right? But if we want to look at any of those sublethal effects, for instance, at low temperatures that don't quite approach that freezing point, then we can use somewhere from 0 to -2, and that's the ones I'm using right now. So we can program the bath to something like 0 degrees or -2, and we can apply the cold stresses in groups of 10 for flies. So we have like a plastic vial. We can get 10 flies, put them in the vial, put a little foam stopper, so all the flies are sitting at the bottom. We can put that vial in the circulating bath. And as long as the vial is in the bath, all those flies are being subjected to that temperature. Then we come back after 5 hours, 10 hours, right? Depending on how our experimental setup is. We pull the flies out. If we want to look at the chill coma recovery, for instance, whether they recover or not, we just take the flies out, put them on a bench. If any stand up, then they recovered. If we want to look at survival or mortality, then we just come back 24 hours later and look at whether, these flies died, 50% of them died, or 10% of them died, or all of them survived, which means that it wasn't stressful enough.
Do you breed any of those flies that underwent cold treatments to see how like their progeny are affected?
There was a study that looked at selective breeding based on cold tolerance phenotypes. So I don't know if they used Drosophila melanogaster or if it was another species of Drosophila, but they did have some flies that were cold tolerant, because there's always going to be variation in the population. Like not every fly is going to tolerate the cold the same way, but the flies that do survive very well, for instance, right? You can take those flies, you can breed them, and I remember that paper finding that the progeny did very well compared to the flies that weren't quite selected for those cold tolerance traits. So there's a bit of a generational effect over there, which is pretty interesting.
I can link the paper in the description. So one thing I should have probably asked you a little bit earlier is, can you just provide us with a bit of physiology of how what the structure of a fruit fly is, just because I want to ask a few specific questions about the effects of those structures as it relates to cold tolerance.
So fruit fly anatomy is very simple relative to humans, I guess, because they do have a brain like us, and they do have a tracheal system, kind of unlike us in the sense that we have our windpipe, we have lungs, right? But their tracheal system kind of extends towards the organs directly. And their circulatory system, they don't have blood vessels like we do. their circulatory system is open in the sense that all the organs are sort of bathed in this fluid called hemolymph.
Mm-hmm.
So we have blood, right? And insects have circulating fluid called hemolymph.
Okay.
And all their organs, their gut, their muscles, their fat body. Their fat body is kind of like the analog to our liver, you can
Mm-hmm.
think of it.
Mm-hmm.
All these organs are bathed in this hemolymph at all times.
Okay.
And what we know about damage that happens in fruit flies has been looked at in the gut, and we know that the gut becomes damaged as a result of cold stress.
Okay. So then, how do they breathe? Because you said they do have a brain like us. I'm not sure what the how the brain functions. I mean, do they have a heart?
They do have a dorsal vessel
Okay.
that sort of delivers hemolymph,
Mhm.
sort of like it's. You can think of it kind of like a tube.
Yeah.
This tube does have hemolymph
Yeah.
in it, and it makes sure to circulate the hemolymph throughout the body, but just
Okay.
having that open circulatory system open
Yeah. Yeah. Okay.
at all
okay.
times.
Okay. Okay. So nothing necessarily. Yeah. I guess
Yeah,
that's
it's
what I
nothing
was-
too fancy, I
Yeah.
guess.
So how do they breathe? They don't have lungs, do they?
They don't have lungs, but they do take in air from the outside anyway through
Mhm.
their tracheal system.
Okay. I remember there was something like studying this from biology. They have like small little holes along their body called spiracles. Is that
They
right?
do. Yeah.
Is that how they take in air?
They can also take in air through the outside as well.
Okay,
And
okay.
air can find its way from the spiracles as well into their body as well.
How do they excrete things? How do they
Mm.
filter things out from their body?
So
Do they have that ability?
they do have they do have a renal system just like
Mhm.
we do. Like we have kidneys
Yeah.
that work to filter any solids from the blood. They work to regulate the osmolality of the blood, like the water pressure and things
Mhm.
like that. They do have a renal system in the sense that the organs called Malpighian tubules, they kind of look like spaghetti because they're
Yeah.
kind of like these really long appendages um, that sort of like extend into the body cavity of the insect.
Okay.
That's also bathed in the hemolymph. But these tubules regulate the flow of sodium and potassium in the hemolymph, and it also works to regulate the concentration of water as well.
Mhm.
So they do have a filtration system in that sense. But in terms of excretion, their gut is very, very straightforward in the sense that it's a single tract organ. So they ingest whatever food they eat, right? It goes into their mouth, and whatever food goes from the mouth, it goes into the first part of the gut called the foregut.
Mhm.
That's the anterior region. You can't think of it like the first part of our esophagus, right? And then anything that goes past the foregut goes into the midgut. That's the central portion of the gut. And then to the hindgut, that's the posterior region. And then it goes from the midgut to the rectum. The rectum is part of the hindgut as well. And then from the rectum, it goes outside of their body. So it's
Okay.
very uniform and like single tract.
Right. Right. Okay. Do you know why they do this? I know we've probably all seen flies do that at one point or another, and I've always wanted to know why.
So
It's like they're preparing for some
it always
scheme
looks
to
like they're
hatch.
scheming. Yeah. So flies typically rub their appendages
Mhm.
together to taste whatever they're on. So
Oh.
they're walking on a surface, so they're
Yeah.
picking up these particles, right? But when they rub their hands, they're sort of like trying to get a taste for the food. That's why if you look at a fly closely, it does this rubbing motion, but then it sort of like makes contact with its face like that as well. Sometimes it's kind of like hard to see because it doesn't always do it, but if you look at a fly, sometimes it does this, but then it does like a motion like that.
Interesting.
Yeah,
They're trying to pick up a scent from something.
exactly. They're trying to like taste whatever they're on. They're trying to get a feel for like what their environment is.
Can I eat this? Yeah.
That's the question on their mind. Exactly.
Yeah. So, all right, back to cold tolerance.
Mhm.
What is the purpose of understanding how tolerant they are to cold, and does that purpose extend to being able to have, let's say, bees, for example, that can tolerate more cold climates?
So that's a good question because a lot of the research that goes on in our field, at least, and a lot of the research that we're familiar with directly, kind of looks at it from a fundamental perspective. So we're always trying to look at how insects deal with cold temperatures at a physiological level or a genetic level,
Mhm.
right? And the other side of the coin is, well, what can we do with this information, right? You can look at it in the sense of pollinators, like bees, for instance, right? And, at the beginning of spring, where we need these bees to be outside in order to get a head start and pollinate all these plants before anything that could harm these bees, like predators, also like follows them, right? some people might be interested in the question of, well, can we make bees cold tolerant? If we can't make them, is there a way to artificially do that, right? If that's the case, maybe you could release these bees earlier than spring, March, April, for instance,
Mm-hmm.
right? And have these bees establish themselves in the outside environment and not have to worry about the possibility of these bees having to face anything that could potentially threaten their numbers early on. You could also look at it from the perspective of pests.
Mm-hmm.
So things like mosquitoes, for instance, which, we don't like a lot, relatively,
Not at all.
compared to other insects like bees. Bees
Yeah.
are cute.
Mm-hmm. Bees are
We
nice.
typically don't think mosquitoes are adorable, right?
We like bees.
Exactly.
We don't like mosquitoes.
Team bees all the way.
Yeah.
But you can look at it from the perspective of pests,
Mm-hmm.
like mosquitoes, right? And ask, well, these mosquitoes may behave the same way as flies do at low temperatures. And is there a way we can use that information to make mosquito populations even more susceptible to the cold by targeting certain things
Mm-hmm.
in their body, right? And so if you're working to either rid a certain region of a specific insect pest that damages trees, for instance, right? Maybe you can apply something to a plant, right, that targets certain things in their body. The insect does eat these plants that have this compound, right, this chemical, which would in turn affect its cold tolerance. If that makes sense.
I understand, but that also raises the question: are those changes going to affect how we eat those plants? Not how we eat those plants, but are they going to affect what happens to us when we eat those plants? Like, does it play any role on us?
Yeah.
I imagine, yeah.
And I think that's always a question that we need to be mindful of, right?
Yeah.
And getting an understanding first and foremost as to how insects deal with low temperatures at a physiological level is something that we need to get a foothold first. Because right now there's a lot of correlation going on
Mm-hmm.
in these studies that pop up saying that, which is not bad in and of itself. This is how science is, right? It always tries to establish certain factors and seeing how these
That's
factors
right,
relate
yeah.
to each other, right?
Mm-hmm.
But if we can ideally get down to establishing causal relationships between low temperatures and injury
Mm-hmm.
and seeing exactly what it is happening inside the insect's body that leads to cell death. Because right now there's a lot of correlation saying that low temperatures relate to ion and water imbalance in their bodies. And that's a driving force of tissue death. We know that because a lot of evidence has been mounting in the past decade or so, saying that if you stress an insect out at low temperatures like Drosophila or locusts, the levels of potassium in their hemolymph go up, and that's a main driving force behind cell death.
Why does that kill? Why does that cause cell death?
They don't know we're recording an episode.
Yeah.
Bad manners.
So why does that cause cell death?
Hmm.
The change, like the imbalance between calcium and potassium.
I want
Is it calcium and potassium?
potassium.
Potassium. Okay.
Potassium in particular. And I want to say that question is one of the reasons Heath started his lab here.
Ah, very good. All right.
So this is what I mean. So a lot of the work that Heath did, as well as other prominent researchers in the field at Western University, at Aarhus University in Denmark, right? They've been establishing this constant pattern that they've been seeing where cold stressed insects, the low temperatures lead to a concentration of potassium in the hemolymph because water concentration goes down.
Mm-hmm.
The volume of hemolymph decreases as a result of that low temperature, and that water goes into their gut. So there's this net movement of water into the gut of the insect, and that drives the concentration of potassium in the hemolymph, which is outside of the gut. It drives the potassium hemolymph up.
Okay.
And it turns out it's a combination of that high potassium environment as well as low temperatures that drive cell death. And this was the reason we know this is because rather than looking at it just from a whole organism perspective, an in vivo perspective, where you take the whole insect and you put it in the cold, right? And we know that tissue damage happens in the muscle. So what was done was that they took the muscle out and they put it in a solution, so an in vitro solution with the muscle tissue. And they just exposed that muscle tissue to a high potassium environment, so they kind of spiked the saline with potassium, but nothing happened to the muscle. It was still healthy. They stressed the muscle tissue with a low temperature, but buffered the saline so that no potassium built up and nothing happened to the muscle either.
Interesting.
So it's a combination of both the low temperature as well as a high potassium environment that is responsible for tissue death. But from a mechanistic standpoint, when you have potassium building up, right, why does the cell die, right? And what are all the steps leading up to the buildup of potassium
That,
outside of the cell? That is what we're still missing right
yeah,
now.
that's what I'd like to know.
And that's kind of like what we're talking about when it comes to like causal relationships. So
Hmm.
we know a correlation exists between low temperature stress and hyperkalemia or
Yeah.
high potassium,
Okay.
but the exact causes, that's what we're still trying to find out.
Okay. Okay. And you said that most of the damage seems to be focused in the gut, right?
The damage does happen to the gut and the muscle tissue from what we know, from what has been done. Yeah.
That damage occurs
Hmm.
during, let's say, a certain amount of time of the insect being incubated in cold temperatures.
Yeah.
Uh, you let the insect start to warm up again so that it doesn't die.
Yeah.
How does it behave afterwards, having
Mm.
sustained that kind of damage during
Mm.
the incubation period?
So one way this has been looked at is by observing the behavior of like a healthy insect compared to a cold stress insect, right?
Hmm.
And you can actually quantify this. You can quantify this from a behavioral perspective as well. So typically, what researchers tend to do when looking at cold tolerance is just looking at mortality. So whether something is alive or dead. And typically, what we define as something being alive is if something is moving.
Hmm.
So if something is moving, right, no matter how much it's moving, it's still not dead, right? You can put maybe like a 1 for something that's alive if
Hmm.
you want to quantify it, and a 0 for something that's dead, right?
Okay.
But the degree of movement that an insect can display can vary,
Right. Yeah.
right? So I'll take flies for instance.
Hmm.
And a fly can walk, right? It can climb, it can jump, it can fly, right? And if you were to assign a score to this, it would be like a maximum score in
Hmm.
terms of like movement.
Hmm.
But if a fly is dead, right? And it cannot move at all, then that would be at the bottom of the scale.
Yeah.
So then you have to come up with a system of everything in between. So if you cold stress a fly and if it's not able to stand up, but it's still like trying to like struggle on the ground or like at the bottom of the container, it's like moving its limbs and stuff, that would be like 1 point above the bottom.
Right.
And then you have to like see like, oh well, if the fly is like standing up but it's not moving, you go 1 point more.
Hmm.
If it's walking around but it can't climb against the force of gravity, then that would be 1 point more, right? All the way up till the top. So we kind of have like the scoring system right now in our lab, and we've been adopting this over the past few years, and this is based off of a similar system that Heath was using about like a decade ago
Hmm.
in locusts, but he
Yeah.
also adopted it for flies as well. So just by having flies stressed out at low temperatures, you pull them out and you let them recover, and then you can track their movement over that 24-hour period
Hmm.
following that cold stress.
Okay.
And then you can see like, oh, maybe like these flies are doing very well at the 3-hour mark after I took them out of the cold. Do they drop down? Do they do just as well? Do they improve? We can use that info, and it paints a better picture. It paints a lot better of a picture compared to just looking at live versus dead, in my opinion.
Okay, that's fair. I'm kind of interested to see if you know, is their ability to digest food or break things down change after the cold incubation?
That's a good question because no one has quite ever looked at this very well,
Hmm.
in the sense that whether they can eat
Yeah.
at all, right? Or whether their feeding is impaired,
Hmm.
or whether their feeding ability is unchanged, but their digestive ability is impaired.
Yeah.
No one's quite looked at that.
Okay.
And that's still something that we've been like toying around with,
Hmm.
but maybe at some point we'll get down to it.
Do they die faster after they've been incubated in cold temperatures? Like, do you
Hmm.
keep them after? So, in your experience, when you
Mhm.
are running these experiments, you're subjecting them to cold temperatures, and then you let them rewarm and try to observe their behavior afterwards.
Yeah.
What is the degree of survivability after that period? Having said that, we're not going to be subjecting them to cold temperatures again.
Mhm.
How likely are they to survive for as long as an insect or as a fruit fly that has never been subjected to cold temperatures,
Mm.
and they're of the same age?
Yeah, so controlling for age, as long as an insect wouldn't go in the cold,
Mhm.
that insect will be alive, right?
Okay, yeah.
So as long as you keep an insect at its rearing temperature, like
Mhm.
25 degrees, you don't, pull it out, you don't put it in the cold, that insect is going to be alive
So
very well.
what's that duration, that timeline that they'd be alive from the point that they're, let's
Mm.
say they hit the adult phase
Yeah.
up until they die? How long is that usually?
Mm. So when we talk about longevity or total lifespan of an insect, there's going to be a bit of a difference in population.
Hmm.
But from the flies that we have in the lab, we've never done an actual longevity experiment with the population that we have, but they live for at least 40 days from what we've seen.
Hmm.
And so long as you have these flies and you keep moving them to fresh food, whether it's every day or every week, they can live for at least a month
Okay.
comfortably. Compare this to a fly that you did take out of the incubator and you put it at something like 0 degrees,
Hmm.
right? If that if that duration at 0 degrees, that low temperature environment, was stressful enough, then depending on how injured the fly is, it is likely to die maybe in like a few days.
Hmm.
So if it did suffer enough injury where you did observe a lot of these motor defects where it
Hmm.
wasn't moving quite well, for instance, during that rewarming period, then there's a very good chance that it might die. And this is something that a student in our lab, Mitch Allen, he does
Yeah.
honors thesis with
Yeah.
us, and he did use the scoring system,
Hmm.
and he did track these flies at 4 hours after they were cold stressed, as well as the 24-hour mark as
Hmm.
well. And interestingly enough, how well a fly moves at the 4 hour mark after it was taken out of the cold, you can quite strongly predict whether that fly will survive or not.
Okay.
So any flies that do show degree of damage where they're like not moving well, it's very unlikely those flies are gonna make it.
Hmm. Now, the ones that do make it after the 4 hour incubation,
Mm.
how likely are they to survive to that complete, or not? Let's say not complete. How are
Mm.
they? How likely are they to survive to that 1 month period?
Mm. I don't think we ever did work where we did cold stress flies and then kept them alive for the remainder of
Hmm.
their lifespan. Because typically when a big reason for this is because the population that we have, so long as we're not using them for experiments, we try to mitigate any effects um to their lifespan by like keeping them in as controlled conditions as we
Hmm.
can, right?
Hmm.
So, if whatever insects we cold stress, we typically just like discard them at the end of the experimental run.
Okay.
So we've never quite looked at it, um, and saw whether their longevity or total lifespan increases or even decreases as a result of the cold stress. From what we know, is only limited to what we've been doing in our experiments where we only track them to that 24-hour mark.
Hmm.
And what we've seen, what Mitch's seen, what I've seen, is that if you cold stress a particular fly and it comes out doing very well, there's a very good chance that it will survive to the 24-hour mark.
Okay. Okay. Understandable.
Yeah.
Now, moving to talk a little bit more about their immune system, because I too did not know that fruit flies have an immune system. What are they? What kind of pathogens do they have to worry about defending against? Like,
Mhm.
are bacteria and viruses present that can infect flies, and that
Mhm.
flies have to worry about just as much as we do?
Mhm.
If not, what are other pathogens? I know fungi probably is
Yeah.
definitely one of the predatory pathogens that
Yeah.
flies would be scared of.
Yeah.
But how does their immune system work?
Mhm.
And how do they use it to defend against pathogens?
So flies do respond to bacteria and fungi and viruses as well. The major difference between the fly immune system and our immune system is that they don't have an adaptive immune response like we do. They only have an innate immune response that consists of these specialized peptides, these specialized cells that work to fight off pathogens directly. So whenever they do have an infection from the outside, whether it's fungi or bacteria, certain components of those pathogens are recognized by the insect's body.
Mm-hmm.
So, in the case of bacteria, for instance, the cell wall, the peptidoglycan in the cell wall, that is recognized by the immune pathway. If it's a gram-positive bacteria, you have a response from one of the immune pathways. If you have gram-negative bacteria, you have a response from another immune pathway.
So, two questions there. One, could you just explain the difference between innate and adaptive immune system? Because we have both.
Yes.
But flies, you said, only have an innate one, right?
Right.
And then could you also explain the difference between gram-positive and gram-negative bacteria?
Yeah. So innate immunity are all the components of the immune system that work to fight off pathogens if they're recognized by the body, right? Compared to adaptive immunity, where a certain antigen is picked up in the body recognizes that, it produces an antibody for that antigen. So when you do get infected with that same thing, you won't get as sick as the first time. Your body adapts to that infection, and it produces this antibody in order to recognize that the next time is picked up, right?
Okay.
But flies do not have that adaptive component. They only have the innate component. So when you think of components in the body, such as white blood cells, for instance, flies do have similar cells called hemocytes. These hemocytes, they're specialized immune cells that circulate in the hemolymph in their body. And if they pick up whether it's bacteria or fungi, they can engulf it via phagocytosis. They can work to engulf it and like sort of like form stacks on it in order to immobilize it if the pathogen is a bit larger or the body is a bit larger. They can also form melanin in their body, where the melanization response surrounds that object and sort of immobilizes it to kill it as well.
So it can form melanin around pathogens to kind of immobilize them, you said? How do they die afterwards? What kills them? Just the inability to eat anything or to sustain themselves?
Yeah, the inability to proliferate, but also if any compromisation is done to the cell membrane, right? And any damage is done to that outer membrane of the pathogen, whether it's cell or the cell membrane, then you have some perforation that happens, and all the components inside the cell become exposed to the outside and they leak out. That leads to the death of the pathogen.
Okay. Can they have any memory to pathogens that's infected them before, or that's the part that they're missing?
So that's the part that they're missing. That's the adaptive component. So you could infect an insect with some pathogen, right? The body will respond by producing these specialized peptides, these specialized molecules to fight it off. But after a period of time, once levels of those circulating components go back to normal, you infect them again, they're just going to show the same thing. So they're not going to do any better.
Okay. And then the difference between gram-positive, gram-negative bacteria.
Yeah, so gram-positive bacteria have a thicker cell wall compared to gram-negative bacteria that have a very thin cell wall on the outside. But the reason I mentioned gram-positive and gram-negative is because the two immune pathways in the fly that produces these specialized peptides, they recognize the type of peptidoglycan.
Okay.
So let's say you have a gram-positive bacteria, right, that has that thicker cell wall. The composition of that cell wall is a different makeup, you can think of it. And that makeup is recognized by specialized proteins that circulate in the insect body. They're called peptidoglycan recognition proteins. You can think of these as sort of like little sentries that sort of like circulate the body at all times to try to pick up anything that's happening, right?
Okay.
They pick up this type of peptidoglycan, and that triggers that immune signaling through one of the pathways called the toll pathway. When you have signaling happening through the toll pathway, you get this secretion of specialized peptides called antimicrobial peptides, AMPs.
Okay.
And it's these AMPs that work along with the hemocytes and the lamellocytes that form the stacks, as well as melanin, in order to fight off any infection in the body. And these peptides, the way we think they work, there's like common models there's like 3 models as to like how we think AMPs work, but these peptides have a slightly positive charge on them. And the membranes of bacteria have a slightly negative charge. So the attraction between these two has the AMP attracted to the bacteria, and the AMP sort of slots itself into the membrane. When the AMP slots itself into the membrane, that creates a sort of perforation or sort of opening in the membrane of the bacteria, and it compromises it, and the bacteria dies.
Okay. So it kind of creates a little entry so that the membrane, what actually kills the bacteria? Does hemolymph get in there that can break the bacteria down, or is it just the bacteria's inability to? What is it exactly that kills the bacteria?
It's like one of the common things of cell death. If you have any compromisation in the cell membrane, right, and all the components are sort of like open into the extracellular space, the components are just going to be released, right?
I see.
And
so it's not actually, so it's a full opening. I thought it was inserting itself and kind of making a hole, but also plugging it at the same time because it's inserting itself into the hole.
Yeah. So that perforation would be large enough to compromise that membrane in its entirety. And so that bacteria would die.
Understandable. Okay. And so then how does the correlation between cold tolerance and immune responsiveness work?
So I think I mentioned at near the beginning that we only know about this from like the past decade's worth of work, right? And what has been shown in the past decade is those cold stresses, those cold exposures, you expose an insect to the cold. If you look at certain immune genes, specifically genes that encode for these AMPs, the expression of those genes go up.
Mm.
Now the question is, well, these insects, they're not facing any foreign pathogenic stress,
Right.
right? There's no bacteria, there's no fungi, right? If they're being stressed out in the cold, why do we see immune genes go up, right? Sterile immunity is not a foreign concept because, you can take us, for instance, right? And let's say you do have, you're playing football, for instance, right? You take a knock to the arm, right? That you notice that local redness, that local swelling that happens, right? You still get this inflammatory response in your body because of mechanical injury. Like you
Right.
take a knock to your arm, right? You're gonna have that localized swelling, right? And sterile immunity does happen even in insects as well. So even if you take an insect and you prick the outside of its body with like a needle, like you make it like a little a wound, right, with a needle, you also get some degree of immune activation. Like you have immune genes go up, you have these hemocytes that migrate to that site of injury to seal the wound in order to like recover that injury, right? Even insects that are exposed to ultraviolet light. This is based off only a few studies that have looked at UV exposure, even heat stress, right? These are all abiotic stressors. Not necessarily biotic stressors that involve bacteria and fungi, but cold is just another one of those abiotic stressors, right? But now the question is, and this is one of the main questions that I'm trying to look at, is the tissue injury that happens in the cold. Is that tissue injury causing immune activation for some reason? What is the relationship between cold stress, tissue injury, and immune activation that happens in Drosophila?
Do Drosophila experience similar inflammation as we do? So like you said earlier, when if you were to prick it with, for example, with a needle on its wing or somewhere to create a wound, does that wound cause an inflammatory response where you get activation of all of these different immune pathways to try it and like you said, fix up the wound and heal it basically?
So there's a really interesting study done in 2020. It came out in Cell Reports, if I'm not mistaken.
Yeah.
But that study looked at how mechanical wounding induces that immune response in flies. It was a very well done study and established a lot of those links, I guess, that we can use even in the context of low temperatures. So what they found essentially was that pricking the cuticle of a fly, it damages those epithelial cells, right? You have this cut that happens, you have this opening, right? The formation of melanin will try to seal that wound back up. And you're gonna notice, if you look at a fly and you prick it, you're gonna notice at the site of injury that the color turns dark. That's the melanin sealing that wound.
Interesting. Okay.
Now, the cells that you damage by pricking it, right? Because this was a non-controlled form of cell death, right? And you have this artificial injury that you induced, you're going to see a release of hydrogen peroxide that happens into the body, so reactive oxygen species, as a result of the stress to the cell that you just made by having that mechanical injury. And hydrogen peroxide, being a reactive oxygen species, is a known immune signaler. And hydrogen peroxide in the hemolymph is picked up by the hemocytes. Those hemocytes are what's circulating in the body to fight off that infection, right? And you have hemocytes moving to that site of injury to sort of like close that opening, right? You have hydrogen peroxide going into the hemocytes and triggering that immune cascade through that toll pathway, which normally responds to gram-positive bacteria. Now, there's no signs of pathogenic infection, right? But that mechanical injury, because of what the hemocytes are picking up in terms of hydrogen peroxide, the toll pathway responds. So if you were to take the whole fly, homogenize it, carry out qPCR to look at gene expression, you're going to notice toll genes go up as a result of mechanical injury.
And toll genes also go up during pathogenic injury, or sorry, pathogenic intrusion, let's say?
Yes. So the toll pathway is activated as a result of gram-positive bacterial infection as well as fungi. So in the case of biotic stressors like pathogens, you would expect toll genes to go up
Mm-hmm.
because of that signaling response, and you do see them go up. In the case of cold and mechanical injury, some toll genes also go up as well, even in the absence of any pathogens.
So then my next question would be, what kind of damage do pathogens do on fruit flies that causes that toll pathway to become activated?
Mm.
Like, is the damage being that's caused by cold damage
Mm.
versus like bacterial damage, for example, is it similar in a way that causes the activation of the toll response to be the same in both cases?
Mm. So bacterial infection would lead to septic injury because of the that system-wide inflammation. You
Mm-hmm.
introduce a bunch of pathogens that are known to be infectious to the fly, you're going to get that system-wide activation.
Right.
What we think is that the fly that's being exposed to the low temperature stress, because it's being administered to the whole body, would also be systemic in the sense
Mm-hmm.
that the whole organism is responsible for. Oh, sorry, the whole organism is responding to that low temperature stress.
Okay.
Now, in terms of exactly how bacteria damage fly tissues, well, some work has been done in the sense of injecting flies with some strain of bacteria, let's say Acetobacter, for instance, which is known to cause systemic infection, Drosophila. But, you know, directly correlating, what tissues are injured versus what genes go up
Mm-hmm.
in that much of a detail is a bit being looked at right now.
Okay.
So we do know that systemic inflammation, in both cases, though, does lead to the release of those AMPs, those
Mm-hmm.
antimicrobial
Mm-hmm.
peptides. If you have large number of bacteria in the body, AMPs will be released, and those AMPs are what work to fight off those pathogens. And the genes for those AMPs go up in both cases, in the case of pathogens as well as cold stress.
Okay, so for once the genes, the AMP genes go up
Mm-hmm.
for cold stress,
Mm-hmm.
uh, have you noticed any effects, like any positive effects that correlate some sort of repair to damage or in
Ooh.
response to that cold tolerance?
It's funny that you asked this because one of my last data chapter in my PhD is looking at the overall effect of AMPs on cold tolerance. So I'm currently looking at that right now because that's the big question in the end, right? Because regardless of the causal mechanistic underpinnings
Mm-hmm.
of what activates the immune system, the end result of immune system activation is the release of those AMPs, right? So if you cold stress an insect, AMPs are released. How does that affect their cold tolerance in the end?
Exactly. Yeah.
So we don't know if in the context of low temperatures, we don't know if AMPs are beneficial in that they mitigate injury to the tissue or whether they amplify them.
Mm-hmm.
And what we do know in the case of Drosophila, interestingly enough, is that some AMPs in the brain, if you overexpress them, they can lead to neurodegeneration.
Really?
So they can be cytotoxic to some capacity if the genes for those AMPs are overexpressed.
Mm-hmm.
Why nerve cells die after those genes are overexpressed hasn't been looked at in those papers. They only looked at whether AMP genes, specifically if they're overexpressed or if they're underexpressed, whether they lead to any changes in the longevity of flies. So some AMP genes do lead to reduced longevity. Some AMP genes do lead to the death of cells in the brain.
That's very interesting.
Yeah.
Which makes me really curious of how these AMPs actually function. You just said that they do insert themselves into like gram-positive bacteria, for example, or
Mhm.
even gram-negative bacteria, right?
Right.
The question here is when they're when they're overexpressed,
Mhm.
is it causing them to insert themselves into like brain tissue, for example? Is that the problem? It's causing like
Yeah.
the same effect in
Mhm.
brain tissue as it does in bacteria, and it's causing them to like basically spill their contents into the hemolymph.
Yeah. See, that's a really good question because the broader picture for my last chapter of my PhD is to look at the overall effects of AMPs on cold tolerance, but also to try to figure out how AMPs interact with healthy tissue in
Mm-hmm.
flies. So let's say if we did have AMPs, right? Let's say if you synthesize these AMPs and you injected them into a healthy fly, even in the absence of any cold stress, right? If
Yeah.
you injected it into a healthy fly, does it show any changes lifespan, for instance, right? If it does, kind of like along the lines of what you're saying, because resting membrane potential of healthy cells is around negative 70 millivolts,
Mm-hmm.
which is net negative charge, right? Compared to the AMP, which has a slightly net positive charge.
Right.
So by that logic, if the AMPs are attracted to bacteria because of that charge difference, you may expect the same thing in healthy tissue as well. where the AMPs are targeting the healthy tissue and perforating the membrane. But we don't know if that's the case
Right, right.
because that has not been looked at either in that study, but at least in the context of low temperature stress, I think it's a natural follow-up.
Work in progress kind of thing.
Exactly.
Okay, it's definitely an excuse for me to have you on the podcast again. I wanna know what's going on once you finish your PhD.
I think that makes it two of us.
Fantastic, yeah. So then a future approach or a future directions, let's say, could be to, after you answer that question, then let's say, for example, that AMPs do have a net positive benefit. Then would it be possible to inject them into other creatures? But I suppose then, I don't know if all insects have the same sort of defense mechanism. So how many other types of bees, for example, do they have the same defense mechanism as fruit flies, where they start to secrete these types of AMPs in order to defend against whether it's pathogens or it's cold tolerance?
So one thing we have going for us, actually, I should mention, is that fruit flies, we do know a lot about their immune pathways, all the genes involved, all the AMPs, because they have a fully sequenced genome. If you're looking at something like a bee, where we're still not as familiar with the body or with the genome, right? We can't quite draw parallels. One thing that all insects have in common is that innate immune system. So hemocytes, for instance, right? Those circulating white blood cell analogs, you can find them in flies, you can find them in crickets, you can find them in locusts, bees, you can find them across all of them. Now, what types of AMPs you can find may differ from one species to another. For example, one AMP that insects have is called secropin. And secropins were first isolated from silk moths, or large silk moths, back in 1985. So secropins were named after the species name of that moth. And that's one similarity, see, that flies have and moths have, but whether bees have the same thing,
we don't know.
Remains yet to be seen. And there's always more AMPs that we're identifying in other animals as well. You know, beetles, for instance, right? Or honeybees or bumblebees or cockroaches even.
What kind of
we're finding out.
What kind of translational effect do you hope that your work might have on the future of this research?
I think, well, maybe I can talk about it in two perspectives. One, if AMPs do prove to be beneficial, maybe that can help us provide some sort of beneficial survival benefit, to insects that we want to keep alive, such as pollinators. Maybe pollinators that are kept in a low temperature storage facility, waiting to be released to the outside, maybe they can supplement that with those AMPs, right? In order to resist any fungi that are out there that could target the bees, for instance, or any bacteria that could target the bees, to give them that survival benefit. Or even release them earlier in the spring when temperatures are still low, so they can establish their population. In the case that AMPs do show some degree of cytotoxicity, maybe they can be used to rid some area of some pests that we don't want, for instance.
Get those mosquitoes out of there.
Exactly. We don't need them.
Yeah. I mean, there was this, so this is a little bit unrelated to your work in fruit flies, but there was a study that I read about a couple of years back where what they were trying to do, I mean, the effects of it have already been demonstrated, where they mutate certain progeny of mosquitoes to prevent them, so that the next generation becomes sterile. Now, I was always curious as to what kind of effect that might have on the ecosystem surrounding it, because I'm sure that, you know, while mosquitoes are not the most fun insects to be around, they're probably food for some other type of insects that might be more beneficial to us. So that's kind of that would be my main concern, is how's that affecting the ecosystem on a more grand scale.
Yeah. Well, I kind of say it in jest, like, oh, we don't want mosquitoes, but in the grand scheme of things, right? Any organism taken out of the local environment will have some sort of downstream effects, whether, as you said, whether any animals are dependent on that mosquito for food, or any of a number of reasons why mosquitoes are important ecosystem players, or where they provide some sort of service to that ecosystem, right? It's important to have them around to some degree, but that sterile insect technique, right? What you mentioned about, you know, sterilizing the males, releasing them to the environment so that there's no progeny. To some degree, you're getting rid of the mosquitoes. Yeah. But also, you're not immediately removing them from that environment as well. So whatever depends on the mosquito for food, for instance, is still going to find it. And ideally, you don't want to disrupt the natural rhythm of the ecosystem all of a sudden
Right.
and have
Yeah.
something immediately removed. Otherwise, you can't predict what might
Hmm.
happen.
Hmm.
And
Maybe give them enough time to kind of adapt to the lessening populations of mosquitoes so that they can have something else
exactly,
to kind of feed
exactly.
on.
Sort of introduce that change a bit more slow
Hmm. Hmm.
and see what happens.
Okay. Well, one last question I'd like to ask you is something that you may have kind of elucidated to earlier.
Mhm.
Why fruit flies? Why study fruit flies? Why work with fruit flies? And why is it the organism that you guys kind of selected to be your model for all of these experiments?
Mm. So one big reason why we use fruit flies is because it's very, very inexpensive to rear.
Mm-hmm.
And typically when if you want to work with insects, one thing that a lot of biologists need to keep in mind is statistical power. When it comes to designing experiments, you want to have a big sample size, right? And when you talk about sample size, having a bunch of fruit flies in your incubator can lead to you having thousands of them at a time. And how long it takes for a fly to go from an egg to an adult is about 10 days at 25 degrees.
Mm-hmm.
If you compare this to other model organisms, if we're studying thermal stress, like locusts, for instance, locusts are a good model to look at when looking at physiological effects of cold stress. Like we know muscle damage happens because of what has been done in locust muscle tissue, but
Right.
it takes about 6 weeks
Oh,
for a locust
okay.
to go from all the way from an egg to its adult stage.
Mm-hmm.
So you're waiting about a month ish while waiting for the locust to be ready for experiments. If I have a bunch of flies, I can move them to a bottle of food, wait 2 hours, move them out. 10 days, I have about 200.
Okay.
It's very expensive, very fast generation times, as well as having a fully sequenced genome as well.
Mm-hmm.
So if we want to look at loss of function studies, so what you mentioned about whether AMPs have a positive or negative effect, um, I have some lines right now that I'm working on that are CRISPR Cas9 engineered, where some genes are knocked out. So you look at the gene on the chromosome, and the gene encoding for one AMP is gone. I want to see how this AMP is important for the cold tolerance. So I don't need to do anything like very fancy. I can just have this genetically engineered fly, put it in the cold, compare it to a wild type with a healthy genome, unchanged, put it in the cold, and compare how the two strains of flies do.
Mm-hmm.
Whether one survives better, whether they both survive the same, that helps to. It's more of a top down approach
Right.
where you're still looking at phenotypes, but it gives a better picture.
Okay.
And especially when you're working with flies, just having like hundreds of them in your sample
Mm-hmm.
size to account for that individual variation in cold tolerance just makes it all the more powerful.
I remember you mentioning that last time we had a chat about the different Cas9
Mm-hmm.
flies that you had different AMPs removed from.
Yeah.
So you're just trying to basically study to figure out which AMPs are really involved in cold tolerances.
Yeah, exactly.
Is there a possibility of some kind of congruent effect where if you remove multiple different types of AMPs, then there's a more catastrophic effect on how they are able to sustain cold temperatures?
Yeah, so some of the AMP lines do have a combination of two or more AMP genes that are removed. I'll give you an example of two of the lines that I have. One of them is missing the extracellular cytokine for the toll pathway. So toll signaling depends on a cytokine called Spatzel.
Mm-hmm.
Without Spatzel, there's no signaling happening in the toll pathway. So that whole pathway is dysfunctional.
Why was it named that? Just out of curiosity, do you know?
I actually don't know why. And a lot of the genes in flies just have weird names overall.
What's the weirdest name that you've encountered so far?
Barbie and Ken. I think it's involved in some sort of like circadian rhythm or something like
Okay.
that.
Very interesting.
Yeah, fly genes have weird names.
Yeah.
Spatzel is one of them involved in the toll pathway. It's a key player.
Mm-hmm.
Without Spatzel, there's no downstream signaling. You don't have the transcription factor being translocated. No AMPs are produced.
Yeah.
And I have another line that's missing the transcription factor for the IMD pathway. This is the pathway that responds to gram-negative bacteria. Without the transcription factor, you don't have AMPs being produced, right? So that whole IMD pathway is dysfunctional. So this is like these two lines are missing a lot of AMP genes. So all the AMPs that are signaled through the toll pathway are gone. In the other line, all the AMPs that are signaled through the IMD pathway are gone. So even just by starting with like these two, right? Comparing it to the wild type, you can see, oh, is the toll pathway important for cold tolerance? Is the IMD pathway important for cold tolerance?
Mhm.
If you do see differences, then you can start to hone in on looking at the specific AMPs in these two respective pathways and seeing whether their individual or combined contribution is
Mhm.
important
Mhm.
in the bigger picture. Because otherwise, if you're working with 18 knockout lines at once, you're looking at 900 flies or so that you're looking at over 24 hours, and it's way too much.
Yeah, that sounds like a lot of work.
Power on
So
numbers.
yeah, that's fair. So then, one last thing I'll ask is, how do you guys actually select for which AMP genes to knock out, or how do you select a combination of the genes to knock out? Is it based off of uh, disclosed trials or pre-disclosed experiments that you guys figured out that, okay, this AMP gene affects this kind of pathway, so we're gonna knock it out and see what happens when it's gone?
So one good thing we have going for us is that in Drosophila at least, there are about 14 known AMPs right now. So in terms of numbers, we don't have a lot that we're working with, which is good. So, we can just look at one AMP, for example, that has been previously shown to have specificity towards acetobacter, like gram-negative bacteria, for instance. If we knock out this AMP, right, which we know is important for the insect's response to infection, we can draw a comparison between cold stress and infection as well. And I do want to give a shout out to Mark Hansen. So Mark Hansen was doing work with these CRISPR-Cas9 lines that I have in the context of bacterial infection over at Lausanne University in Switzerland. And I just asked if I can have some of the stocks. So he shipped them over to me, and that's what I'm working with. I'm working with his lines right now.
That's kind of him to do, I guess.
Thanks, Mark.
Well, Mahmoud, thank you very much for coming on the podcast. I really appreciate your time.
Thank you so much for having me.
I hope we all learned something about Drosophila. And I hope that you can find the answers you're looking for in your PhD, and look forward to talking to you in the future to see where your research has come along.
Thank you, George. I look forward to it.
Thanks a lot. Thanks for coming.
Thank you.
Thanks for listening, everyone.